Hey everybody, sorry I wasn't there for class to present this, but better late than never. So me and Dylan had Staphylococcus aureus, and that is an example on the screen of what ours actually look like under the microscope. So the characteristics, it is found in the environment, which would be the soil, water, and air. Most of the time it's found in the nose and skin of humans. Staphylococcus aureus is a gram-positive, non-spore-forming spherical bacteria that belongs to the staph group. So in 1884, Anton Rosenbach, a German surgeon, actually isolated two of the strains from staph, and he named this bacteria for the pigmented appearance of the colonies. Staphylococcus aureus, from Latin word gold. It's actually called the gold staph. And so the two strains that he isolated it from was the Staphylococcus albus also, which now has the name epididymis, and it's from the Latin word for white. So it's pretty common in the medical field. You hear about it quite often. Usually when you go to the doctor's office or even the hospital, they'll ask you if you have a history of having staph. It's long been recognized one of the most important bacteria that causes disease in humans. It is the leading cause of skin and soft tissue infections. Although most staph infections aren't serious, this type of staph can cause serious infections such as bloodstream infections, which also means sepsis, pneumonia, or bone and joint infections. So it does play a role in agriculture. If you look at the picture, on the left, the picture A is a normal mammary gland of a cow, and then option B would be a cow that has been infected with staph. And below those, it shows the different, what it looks like under the microscope from being normal and then having staph. The ability of this bacteria to move between humans and livestock is due to the specific characteristics of the bacteria, as well as modern agricultural practices. While humans appear to be a primary carrier of this bacteria, but with a continued expansion of the livestock industry, globalization, and frequent use of antibiotics has allowed the bacteria to adapt to the environment. So it can go back and forth, which is kind of interesting. So it is also important in the food industry. The picture is kind of hard to see, but this one is a dirty kitchen, not being cleaned properly, so there's more likely to have contamination. And then the below picture is because most infected foods are from poultry and uncooked meats such as ham or corned beef. It's highly vulnerable to destruction by heat treatments and almost all sanitizing agents. So if it's properly cleaned, your kitchen, you shouldn't have a problem. So if you have poor sanitation or restaurants that have poor sanitation, that's how it's easily spread. So some diseases that are caused by this strain of staph would be sepsis, which is a bloodstream infection. This occurs when the bacteria enters the bloodstream. Pneumonia, this occurs most often when the patient has an underlying lung condition. So asthma, COPD, that kind of thing. Endocarditis is an infection of the heart valves. This can lead to heart failure or stroke. Osteomyelitis, which is the bone infection. This can be caused by the infection traveling in the bloodstream or put there by direct contact, such as trauma or IV drug use. So for treatment, for medications, there's oral and IV antibiotics. supportive care, and then medical procedures. Sometimes if the infection's so severe, they may have to make an incision to drain it or even go into surgery to remove the infected tissue. So these are our examples, mine and Dylan's, of our in bacteria while we were in the lab. This was our blood agar. We had a halo around the 3 that we had indented into the agar. This was pretty interesting because you could actually see the halo. It was very clear. And this was after 24 hours. So this is temperature requirements. As you can see, the 25 and 37 degrees, they're both almost alike. They're very cloudy. And then the 5-degree was clear. So our bacteria did not like to grow in the 5-degree, but it can grow in the 25 or 37 degree. Next is pH requirements. Interesting fact about our bacteria is that it can grow in any pH, which also means that our bacteria is hard to kill. Which you've heard anything about staph that is, can be very dangerous and sometimes antibiotic resistant. So it is very hard to kill. The bacteria had no reaction to the ferric chloride. It does not have the enzymes for the reaction for phenylalanine deaminase. Probably did not pronounce that right. But so if it would have reacted, it would have had a different color there other than that kind of goldish color. The citrate test, our bacteria was negative. It does not use citrate for energy. So kind of stayed the same. If it would have been positive, it would have been a very bright blue. There was no dark brown or black noted, therefore our bacteria was negative reaction for the esculin hydrolysis. And say some of them that were positive were very, very dark. So you could tell right away that ours was negative. Motility and hydrogen sulfide production. Our bacteria is negative for motility and negative for H2S production. Oxidase test. After adding 10 drops of the oxy reactant, it did not turn into an indigo blue. Therefore, this is a negative reaction to the enzyme. You can kind of see ours is blue, but definitely didn't turn that bright indigo blue. So our population count. Other cereal dilutions were too high to count. In the 10 to 6, we counted 68 colonies. I say in the first 2, we definitely had way more than 300. And then in the last one, we had fewer than 30. So we went with this one. For catalase test, after adding the hydrogen peroxide, it started to bubble, which means we had a positive reaction. Our picture we kind of took late, so it's kind of hard to see the bubbles, but there was a few there when we got the picture. After inoculating the phenyl red glucose, sucrose, and lactose with our unknown and incubating it, the liquid turned yellow with no gas, which means this is a positive result. So as you can see, all of our tubes were yellow. So the litmus reaction, the liquid stayed the same without much change and only had a small amount of bacteria on the bottom. This result is alkaline. So as you can see on the bottom, there's just a little bit of residue from the bacteria, and it stayed the same color, pretty much. So the nitrate reduction test, when adding both reactants to the liquid, it changed to a red color, which means the result was positive and the bacteria uses nitrate. Oxygen requirements. Our bacteria can grow with or without oxygen. So as you can see, it had a little bit of growth there in the middle. And this is called facultative anaerobus. So getting on to some more interesting things. Fun facts about our bacteria, Staphylococcus aureus, it's also called the golden staph, and aureus is named from its golden color. It can often be prevented by washing your hands, which brings you back to how it interacts with foods, is because sanitation. And up to 20% of the human population may be a carrier of this bacteria. So challenges we faced in the lab was definitely getting used to all the lab equipment and what it was used for. Neither Dylan nor I had any experience with that, so I think once we got the hang of it, it wasn't too bad, but it was slightly overwhelming at first. Figuring out the microscopes and how to best observe our bacteria, that was definitely a challenge at first. I don't even think we accomplished it close enough to the end. We definitely could have still used practice, but once we seen it, it was pretty cool to know that we had accomplished that. And then being confident in what we were doing. So the first day, of course, I'm pretty sure all of us were nervous and didn't know what we were doing. But by the end of it, we definitely felt more confident and were able to move quickly along in the task and not take as much time. Some exciting moments we experienced was completing the gram stain for the second time and being able to see our bacteria. So the first time, we had left the alcohol on a little too long, so it had taken our bacteria away, so we were unable to have a result. But the second time we did it, we had it figured out. And then figuring out our mystery bacteria. So once we seen it on the microscope the first time, we kind of had an idea of it was staph. We just wasn't sure to which one it was exactly, but we were able to narrow it down and figure it out. So overall in the class, at first it was nerve-wracking. It makes you wonder if you really know all the information for your career. But now we feel like it is interesting, kind of puts the pieces together regarding the disease process and then where the problem originates. So when you get that call from lab, you kind of have an idea of what they're talking about, not just kind of clueless. So we discussed it, and our favorite thing about the class was seeing a change or reaction in what we had done in the previous day. So every day we came in and seen something different or how something changed, and just knowing that we had put that together and we were changing it to learn new things was very exciting and helped us feel more confident and know what we were doing. And this was done by Dylan and myself. Thank you guys, and again, I apologize it was late.